Okay, thank you. I have put into the chat for all of you a document, a Google document. This, not this one, sorry. I don't think this one. Should be this one. And this gives you all of the references that I'm going to mention in the talk, so you're not trying to scribble things down while I'm talking. All right, so this brain imaging data structure, I'm going to talk about what it is and why you should be interested in knowing something about it. BIDS, the brain imaging data structure, is a well-accepted protocol. And what it does is it names and organizes neuroimaging data. It was introduced in 2016 in this paper in Scientific Data. There were 27 authors from a broad range of institutions that supported the project. And since its introduction in 2016, it's steadily gained traction in the neuroimaging community. And as a sort of a standard, a convention for sharing data. It's got associated with it several containerized applications, which are basically really easy to run once you've got things into this format, this convention. All right, so the problem that we've had for many, many years in neuroimaging is the lack of a single convention for organizing and describing MRI data. So people, they organize and name their subjects however they want. And they make subdirectories that make sense to them, and they name the files the way that they want. And that's fine until you want to share it with somebody else. So it creates problems if you have online neuroimaging databases of shared data, and everybody's got a different convention for how they name their stuff. It makes it difficult to share and reuse the data because there's all this documentation you have to do to explain how you organized your stuff. And it really makes it hard to do anything like automating pipelines, because if somebody else has named things differently, then they're going to have to rewrite things to get it to work with your scripts. So in more detail, without BIDS, data processing requires you to write custom scripts for each of your processing tools. Done this for years. Scripts indicate what data you're using, or what parameters to use, etc. And you have to decide how to name and organize your imaging data. Alternatively, you have to reorganize your data to fit somebody else's naming scheme. So program X or Y or somebody's special script assumes a certain naming scheme, and you want to use their script, so you have to rename, reorganize all your stuff to make it fit with that, so that you can use their tools. Furthermore, there's other information you have to configure, not just the names of the images in the directories, but for diffusion imaging, for example, there's these text files, the BVALs and the BVEX, that contain information about how you gathered the images and what kind of images they are. And there's different conventions in different processing programs for how those look, whether they're columns or rows, and exactly how they're organized. So then you need to alter the BVALs and the BVEX, for example, for whatever program you're using, not just whatever lab and script somebody has. In addition, some processing steps in tools require that you include metadata like dwell time and phase and code direction. And that means you have to get that data out of the MRI protocol, out of the DICOMs, better remember to do that because a lot of people don't. They're given the nifty data, and the nifty data has already removed a lot of that information from the header. So it's gone unless somebody was good about recording the protocol in the first place. So to share your data, you have to document how you've organized it, how you processed it, and how you stored it. If you want to share it in some public forum, you're going to have to explain what you did. And if someone else wants to apply the same steps you did, then they have to name their data the same way. In this sort of a scenario, automation is really idiosyncratic and it's fragile because it depends on following the naming rules laid out by one person or one lab or one processing tool. And it depends on having a way to document crucial metadata so that it doesn't get lost just because Nifty doesn't record. BIDS addresses all of these problems. So the brain imaging data structure provides a convention for organizing and describing neuroimaging data and its metadata, like the dwell time and that sort of thing. BIDS meets funder guidelines and requirements for data sharing and reproducibility because it tells you how to organize your stuff. BIDS relies on common image formats like Nifty and naming strategies that are probably similar to what most people already use in their labs. So for each image in the BIDS setup, you retain the image metadata, like the dwell time and the phase and code direction, in a separate JSON sidecar text file. And that metadata, as I've mentioned before, would otherwise be lost by the conversion from DICOM to Nifty. So that sidecar metadata is nice because it's human readable and it's machine readable. So it's directly available to you and to the processing software. So it's not just the case that it's as good as having the MRI protocol where you can look through the PDF and find the dwell time for some particular image sequence. It's better than that because it's a text file that your BIDS programs can read. They can look inside there and see what the dwell time is and never bother you at all about it. So this BIDS convention and the retention of metadata makes it easy to automate processing. So a lot of interesting fallout from this. In addition, it makes it possible to use containerized software like Docker and Aptainer. And the containers guarantee the kind of consistency and reproducibility in processing that we would all like to see. Because there have been problems, right, with running a tool like FreeSurfer on your Mac versus running it on a Linux machine versus running it on the HPC. And even if you haven't switched versions of the FreeSurfer software, you get slightly different results because the operating systems underneath are doing different things. So this containerization helps with that. So by providing a single convention for organizing and describing MRI data, BIDS solves the problems I described earlier, right? Data processing is facilitated because each BIDS app knows exactly how the files are named and organized. Each BIDS app can also use the metadata stored in the JSON sidecars. Data sharing and reuse is fully supported, and that's becoming more and more important for all of us. So there's no need to explain to anybody how you have organized your data because that's standardized if you got it in BIDS. Anyone can run the same containerized BIDS application that you did and get the same results that you did. And it doesn't matter what machine they're running on because the containerized app encapsulates a sort of an operating system. So once you've got your data in BIDS, you can easily try different apps and compare results because all the apps understand the same unified protocol. The automated containerized pipelines are really robust and portable and reusable. So all the BIDS apps, and I'll talk about some of them, have similar command lines and can run on all major operating systems and in the cloud. and on your HPC, so portable and reusable. The available apps are extremely useful, sort of basic things, and there are more apps being developed all the time. And ultimately, you lose nothing, okay? If you really want to do some idiosyncratic processing steps, and that's not covered by any BIDS apps, who cares? Somebody's got should turn off their mic, I think. All right, so just because you've organized stuff into this BIDS convention doesn't mean you can't do anything you want with the data that you've always wanted and done before. All right. Thoughts, questions before I tell you about BIDS apps?
Yeah, Diane, I have a question. Is there like I'm trying to think what my question is. Is there a way is it easy for people to get their data into the BIDS format, like if they get it off a scanner, or is that something they have to like have custom software for based on how they get it, or?
I will talk about that because that is a pain point. It has been a pain point. It is less of a pain point now. So there are some good tools for doing that conversion. Some of them don't require any really programming knowledge at all. And some are, more flexible and more capable, but require that you write some Python and several things in between.
Nice. Thanks.
It's definitely possible. All right, so these mature BIDS applications are one of the main attractions of adopting BIDS. The containerized apps that offer almost like plug and play functionality. Once you solve the problem of getting your data from the scanner format correctly into the BIDS format, then you just go. And these containerized apps understand the data structure and off they go and they do a great job. They continue to evolve and proliferate. And in addition, existing neuroimaging tools like SPM and CON are increasingly able to accept BIDS compliant input. So even if they're not offering you a BIDS app themselves, they've come to realize that it's important for them to be able to read data that is organized in this fashion. Okay, so here I'm going to showcase 4 apps that I use regularly and that I use to teach. BIDS and IMAs for defacing. MRI-QC generates quality control reports. fMRI Prep does fMRI pre-processing for you, and QSI Prep does DMRI, or diffusion MRI processing for you. BIDS and IM does both anonymization and defacing of the data. It retains the original data in a source data subdirectory. But once you run BIDS and IM, then you can upload your data set to somewhere like OpenNeuro. You have done the job that all the funders would want you to do for preparing your data for sharing. As soon as you run the defacing on it, it's ready to go. MRI-QC is a quality assessment tool. It does very nice reports for assessing the quality of your BOLD, that is fMRI data, and your structural data, the T1 and T2 images. They seem to be working on also doing assessment of diffusion imaging data. Haven't really seen how that works yet. But very pretty reports. And they've carefully chosen a bunch of appropriate quality metrics that each provide, some unique and important piece of information about the data quality. And so it's an all-in-one solution, runs very quickly. fMRI Prep pre-processes structural data for you, like your T1 data and your fMRI data. It produces what it calls analysis agnostic output. That is, that output is broadly compatible with tools like SPM or CON or FSL or the Human Connectome project. So it'll generate surface formats for you. It produces thorough reports and an extensive selection of confounds that you can use in subsequent analysis and design matrices. QSI prep is like fMRI prep, but it's for diffusion data. So conveniently, if you have run free surfer tissue segmentation in fMRI prep, then QSI prep can use that same output, which saves you computing time. QSI prep also offers a broad variety of really interesting reconstruction approaches for diffusion imaging data. So once you've done that pre-processing, you can do like another layer of output to produce connectomes and bundles and different kinds of scalar measurements. So those are the apps. Any thoughts or questions about the apps? Those aren't the only apps. There are many, many dozens of apps. If you go to the document that I gave you, it'll take you to the BIDS site and all of the many, many apps that they have listed, and then you'll find other ones. When you go out on the web, you'll find that there are, you know, various tools out there that are optimized. Good point, Tom. All right. So I want to give you a quick tour of the BIDS specification. This is so that you will recognize the structure when you run into it and roughly know what to expect. There's a lot of details, and I'm not going to dig into those because you wouldn't remember them anyhow. So there are several points to be aware of. The BIDS protocol specifies the expected directory and file naming that you need to use. Each Nifty file that you have has to be paired with a JSON sidecar file containing metadata. The structure of the JSON sidecar is carefully specified. There's an online BIDS validator tool to help you validate and identify problems in your data set, so you can see whether it's okay or not. You can specify files for the validator to ignore, which gives you additional flexibility. And converting from DICOM to BIDS correctly has been the major pain point, though there are some pretty good tools, and I'll describe one called Easy BIDS for you. All right, so the BIDS data directory contains the unprocessed or minimally processed data that, for example, might be defaced and anonymized with something like BIDSanim. And in this example here, I've called that data directory Nifty. You might call it raw, you might call it BIDS, you might call it Fred, you can call it anything you want. They don't care what you call that top level directory. What they do care about is what you call the directories underneath it, but we'll get to that. Processing results are saved separately from that Nifty data in a directory, which you can call anything you want, but typically people call it derivatives. So derivatives could be a subdirectory of Nifty, but I think best practice now is to make it a sibling or just a separate directory, as illustrated here. Okay, so here's a BIDS data directory containing one subject with a session. The required descriptive files up here, changes, readme, dataset description, those have to be there and they have to be named this way. Directory names for each modality are set in stone. So there's a directory called a NAT, that's where T1 and T2 goes. There's a directory called DWI, that's where your diffusion imaging goes. There's an FMAP for your field maps, and a func directory for your bold. If you don't have one of these categories, you don't need it. If you don't have any bold data, then you don't need a func directory. But if you do have it, you better have a directory. You better put it in a directory named func. In each case where there's an image, where there's a ni.gz image, there is an equivalently named JSON file that goes with it. Okay, so you'll see that structure in any BIDS data set. And that, again, is parsable by both humans and machines. You know what to expect. The names of the images themselves are specified. And the most important thing is that dashes or hyphens and underscores are used differently. So you use a dash or a hyphen to join a key value pair, and then you join together these key value pair entities with underscores. So this is a valid name, sub 219, because it's a subject, so that's the key. 219 is the value of the subject, that's its name. Here's one named Fred. There's a session here, so ses is the key, that is what you have to use. The dash can be followed by any value you want, some string that specifies the name of the session. And then finally, the modality of the file itself.
But the session name shouldn't include dashes and underscores, I assume.
Right, that is true. You should avoid using dashes and underscores inside the names of the things that are going to be values. Good point. All right, so for each of these nifty images, there's a companion JSON file called the sidecar. This is in JSON JavaScript object notation, which is a standard plain text format used on the web, readable by people and machines. And it contains that metadata that I was talking about earlier, like dwell time, which you would otherwise lose. So I'm going to take you through what one of these JSON sidecars look like. This is a BIDS JSON file for an fMRI sequence. At the very least, the JSON file contains information you want for your methods section, like dwell time. But BIDS apps can also read these values, and importantly, much of the information in the DICOMs, that doesn't make it into the Nifty is here. All right, so here's how it's set up. There are keys and values. They're nicely encoded in blue and red for you. There are fields which are able to be nested. So for instance, slice timing information is a nested field, which contains all kinds of information about the timing of your slices in each volume. And the metadata required for processing that I keep harping on. There it is, effective echo spacing, total readout time, dwell time, right there forever in your data set for anybody to use, and any program to use. All right, once you think you've got your data into the BIDS format, you can check it with this BIDS validator online. It gives you the impression that it's going to upload your data, but it's not uploading your data. It's just uploading sort of metadata about your data and checking that things are right. See, so to validate your data, this is what you do. You choose a file, which in this case is actually your data directory, your BIDS directory. You say upload, which it doesn't really do, and it claims it's going to upload all this, but it doesn't. It's just like the immediate thing. And then it gives you back this nice report that tells you where there are errors in your data set, and what you need to do about it. And you can download an individual error report to look at and work through your problems. So very quick and pretty reliable.
Diane,
Yes.
if you have ASL data that doesn't fit into one of those imaging categories, where do you put that? Is there
ASL data does now fit. You put
Okay.
it in a directory called perf, P-E-R-F,
Nice.
perfusion. And there are some extra hurdles with it because not all the information is easily available and extractable from the DICOM. So sometimes you need to add some information. So ASL is still a bit of a special case, but there is a BIDS standard for it.
Cool. Thank you.
All right, converting DICOM data into BIDS correctly has been a major pain point. I think Easy BIDS is probably the nicest tool you could ever want for doing this kind of DICOM to BIDS conversion. It's available online through BrainLife, so it's secure because you're going to upload your DICOM data to it, but that's fine. It provides a graphical interface and it intelligently detects the DICOM types and guesses what they are, and then it asks you a few questions and off you go. So it's pretty reliable. If you're doing really weird new sequences, you're on the edge, then Easy BIDS isn't going to manage it all for you. But if you're doing pretty characteristic normal sequences, Easy BIDS is really good at this. Let's see. Okay, so you start out, you drag your DICOM directory onto the interface and it uploads it. When it's done, it says analysis complete, which just means that it's looked at all the DICOM data and it thinks it knows what nifty files it should generate and what bid structure it should generate. Then it wants you to, and this is optional, generate a data set description. It's nice to put in some kind of a name for your data set at least. You might put in this other information, or you could do that later if you wanted. It's just going to produce a text file. It's going to ask you how you want to name your subjects. And so you can tell it in here, tell it that I've got a session and what I want to name that. And then it's going to do the series mapping thing where it identifies fields, sequences where it has questions for you and it wants you to verify. So here we look at this first one, the func bold, and it says it thinks this is a bold because there's the word rest in there in the series description, but it wants you to confirm that that's right. And then, then you'll have to deal with each of the other little blobs and they'll ask you the questions and you'll go through. And when you're done answering those questions, you get the option to deface the data. And then you can download it or send it to OpenNeuro or send it to BrainLife. So that's a very nice way to go.
May I ask a quick question, Diane?
Yeah, sure.
So this is an online tool, EasyBids,
Yes,
and you said it was fairly secure.
it is. They swear up, down, and sideways that it is secure. Yes.
Okay, do you find you have to like list in IRB forms that this is a tool you're going to be using since it goes onto the web? I don't know, just thinking about
there's
subject
a paper
privacy.
now, which I think you could probably just quote, and you could just point to the paper and say, look, this is a secure online conversion utility guaranteed
Okay,
by such and thus, and here's the paper that supports it.
okay. And they don't keep any copies of stuff.
No,
So, okay, that's
I mean,
cool.
I think your data stays up there for a little while if you want, but you have a lot of control over whether or not and how long it stays. The thing is, you can upload huge amounts of data if you want, and that may take a while to upload and a while to process.
Okay, thank you.
Yeah, there's one other thing, which is that he's implemented this now as a set of containers that you can download to your local machine and run locally. So if you're really feeling freaked out about the upload to BrainLife, then you can explore, implementing this tool locally. And I mean, there are other ways to convert, but I'm not going to get into those. All right, the future of BIDS. I'm going to talk a little bit about BIDS extensions. And I looked up some that are related to statistics, just for you all. And then maybe we can talk about the future a little bit. So there's a great paper I read before putting this together for you all, and I think it's in that document. It's a Poldrack 2023. It's a preprint, but it kind of goes through the history of how they developed BIDS and what their issues were and where they're going next. Okay, the initial BIDS convention was focused on MRI data. But even before its initial release, it was clear to everybody that there were going to need to be extensions to this. Like, you know, originally they didn't support ASL, for example. And at this point, the BIDS convention has been extended to a lot of other modalities. MEG, IEEG, EEG, genetics data, qMRI, which is quantitative MRI, ASL, which is perfusion MRI, or, microscopy data, some PET data, NIRS data, and there are lots and lots of existing extension proposals that are in development. Okay, so of more interest to this group are extensions related probably to things like statistics and connectivity and dimensionality reduction. And I provide links in the accompanying document, but once you go to those links, you can find more. So here are some examples of projects spawned by BIDS and sharing the same general goals of promoting these FAIR principles, that is, making data findable, accessible, interoperable, and reusable. The BIDS connectivity project includes, tries to define a standard for defining connectivity, whether you're using data from structural data, like maybe cortical thickness data, or fMRI, you're doing functional data, or DMRI, so you're doing some structural data. I guess PET and EEG and IEEG and MEG all can produce some kind of connectivity data, and they want a way for those to have a standard way to express those so that it can facilitate analysis and sharing. FitLens will take a BIDS data set and one or more derivative data sets along with a BIDS statistics model file, and so it'll run basically a GLM on your fMRI data using a standardized format, the BIDS stats model. So any of you who have done fMRI analysis, you know that if you do it in FSL, you specify the design matrix one way. If you do it in SPM, you specify the design matrix some slightly different way. Every tool, AFNI, has its own way of doing it. So they want one way to express these models, store these models, that's shared by all different kinds of tools, and FitLens is an example of this. I should point out that MRI-QC and fMRI prep and FitLens all come out of the Poldrack lab at Stanford, and they are in sort of a major force in continuing to support and push BIDS. NeuroScout. NeuroScout is cool. I read a whole paper on this because it was mentioned in the Poldrack article. And what it does is it's kind of split off from BIDS at this point because it's its own thing, but it's definitely totally inspired by BIDS. It provides a platform for fast and flexible reanalysis of naturalistic fMRI data using the FitLens and the BIDS stats model. So what's unique and interesting here is that this is naturalistic fMRI data, which means you put somebody in the scanner and you read them a story, or you have them watch part of a movie, and you record what goes on in their brain during that time. As you might imagine, once you want to analyze data like that, you need to categorize what was happening at any point in time during this naturalistic data. And there are all kinds of dimensions that you might slice that on. You know, was something loud happening? Was something bright happening? Were there human faces was there human language? What words were being produced, if words were being produced? Were there written words? All kinds of things that you might want to look at in data like that. So it's incredibly complicated, right? So what the NeuroScout does is it's collected together a bunch of these naturalistic fMRI studies with, you know, the data from the people's brains and the data from the movies or stories or whatever they were being told. And then they use machine extracted features ranging from low-level perceptual features like brightness or loudness to visual categories like buildings or landscapes or tools or faces, various language features, etc. And the machine goes through and it identifies all of these things in at least some consistent way, right, for the particular task. And then you come in later and open up the NeuroScout data and say, wow, I really want to study across all of these different naturalistic experiments, what's going on when human faces appear. And you use NeuroScout to identify that as the feature of interest, and it provides you with the data that you need to then run the analysis in a Docker container. I haven't done this, but it seems really cool. All right. Then the last tool that I looked at, and I have to admit that I didn't, this is a little above my pay grade, this Model Exchange and Convergence Initiative. So it's a project that aims to develop a standard format for exchanging computational models across diverse software platforms and domains of scientific research. So I gather that sometimes people are writing models in PyTorch and sometimes they're writing models in NeuroML, and you know, never the twain shall meet. Converting from one of these tools to another is a pain. And so this aims to derive a shared model structure representing the models as sort of graphs with nodes and edges that can be translated from PyTorch to NeuroML or vice versa, or you know, whatever your favorite computational modeling platform is. All right, so this is a very fine standard. It's well accepted at this point. I probably shouldn't call it a standard. I should call it a convention. Just names and organizes your neuroimaging data, you know, at the very least, and which means they're ready to submit to public repositories, which we have to increasingly do. And it means that these wonderful apps are available to work with them. And the containerized nature of the BIDS apps simplifies the processing. And a lot of this means that you get to spend your time learning the principles of what kinds of questions you should be asking and what kinds of choices you should be making in processing, rather than spending your time on the minutia of, you know, going back to the scanner tech and saying, I can't figure out what my dwell time is. And, or, you know, or your IT professional, and or which is probably you, saying, you know, FSL won't install correctly. I don't know if it didn't download right, or you know, if it got corrupted somehow, or if this doesn't get along with my operating systems. You don't have to spend your time on that stuff, because you should be spending your time thinking about what the processing does and what the analysis can do. As for the future, I think BIDS has to, and Poldrack points out in the 2023 preprint that they probably need to rein it in a little bit, decide where they want to stop. I mean, they've extended it to genetics and microscopy and PET, and at some point you can't expect it to cover the whole world. It'll get too complicated and be too much for them to manage, financially and in terms of just keeping notes. But it's part of an important effort to make things standardized in a way that's remains flexible for all of us, but hopefully resolves some of the day-to-day problems of naming things and keeping track of information. And that is all I have to say.
Well, let us thank our speaker. And thank you. That was a wonderful talk. Does anyone have any questions for Dianne? We have a few more minutes. Feel free to just speak up. I can't see all of you at once, so.
Yeah, I have a couple of questions. Thanks. That was awesome. Especially I've been sort of like overwhelmed by the BIDS format and like how to even like how I would use it. So it's helpful to have this overview. I'm wondering, as I have a bunch of questions, but like I work in sort of group level analysis. Are there BIDS apps designed to sort of use the format of the derivatives folder that then would be used to do like group level analyses?
That's coming. That certainly hasn't been their main focus to this point. The focus has been getting the solid pre-processing done. But there is a BIDS extension proposal for how the derivatives should be organized, and beyond that, there is this fit lens and the BIDS stats model. I have not tried these, but these come out of the Poldrack lab. They pretty much assume that you have run fMRI prep to prepare the data. So hopefully they're, robust to other choices. But given where it comes from, the idea is you run fMRI prep and then you can use these fit lens, the fit lens tool and the BIDS stats model. I think it limits you to doing GLM, right? But more to come hopefully on this.
Okay, yeah, I'll check those out. My other question was about like if you have or if a repository shares non-imaging variables, is that typically stored in that participant CSV, or is there like a convention within BIDS that describes how to share the non-imaging data?
There are plenty of conventions, probably way more than I can keep track of. There's a huge document online. But yeah, basically, if you want to share the demographic data, like age and sex and I don't know, status on how well they did on some exam or something, all those things can go into the participants.
Okay,
CSV file.
thanks.
Sure.
So I had a question that's a little bit tangential to what you're talking about, but I think is important, which is I'm curious about how, if sort of how the community managed to get their act together to do this in the first place, because there's many areas of science. I mean, I work in agricultural imaging and we face the same types of challenges. And it's just very hard to get everybody on the same page in terms of what the format should be. Everybody wants to share, And so I think there's an agreement that everyone wants to share and everyone wants to make things interoperable, but it's tough to get everybody to agree on
Yeah.
even a standard. And then there's the added complication that there are many certain areas of ag imaging are quite young, and there's a lot of kind of new tech coming out all the time. And so if you fly a remote vehicle or something, drone across your fields, there might be 6 different drone manufacturers and they all use different formats. So I'm just curious about sort of where this came from and like how the community got this launched.
So definitely go and read that Poldrack 2023 preprint paper, because that is
Okay.
what a lot of what he focuses on is how did we get started with this? Where did we make mistakes? Why did it work for neuroimaging? That kind of thing. We did have a lot of different image formats early on in neuroimaging, but the NIfTY format, was sort of the first attempt at really standardizing, and that has had an important impact. Not that the other formats don't still exist, but NIfTY's kind of rules the roost. And I don't know, in some ways neuroimaging was ripe for this, because like you, we needed something, some way, some standardization, but there was also some luck involved. There were some people like Chris Gorgolowski who really, worked on it, spearheaded it, and put a lot of his effort into it. But they also continue to try to make sure that their documents are accessible on Google, that any of us can go in and say, hey, we hate this BIDS extension, or why don't you do this instead, or whatever. So they really try to invite people to participate. They're very good about that. But read his paper. That
Okay, that's great. Yeah, I will. Thank you. Thank you very much. It's very helpful. Yeah,
But
that's
one-
great.
But one thing neuroimagers had working for them that you may not, Dr. Clark, is that there are probably only a half a dozen different scanner types in the world.
Right.
Standard companies, you haven't got 6 new drones coming out every year,
Yeah,
6 new scanners.
yeah, that's true. And I think, that's also been true. That was true, in the in genome sequencing too, right? There's only a few sequencing companies out there. But and so you're absolutely right. But I'm thinking that, maybe one way to approach it is instead of trying to do like all of imaging in ag or all of, at least pick an area where at least there's good buy-in and see if we can get stuff moving. I mean, we've done that a bit in phenotyping and it seems to have been successful. So I think it's one of these things where you got to start somewhere. And hopefully if we can get good buy-in by the community, then then maybe the next drone manufacturer will decide, oh, there's benefit to me to offer the data in a way that is amenable to this format because that's what the community likes to use, right? So there's kind of this back and forth between the manufacturers and the community, and it's like, unless we start getting ourselves together and then saying, hey, if you want us to be involved in your hardware and your data acquisition, then you got to make it in this format. So maybe we can, push a little bit on solving the, a different drone every 6 months problem. But I appreciate that. Thank you, Tom.
I should say that despite there being only a few MRI manufacturers, that nevertheless something like as simple as a T1 image from one scanner will have different features than from another scanner. And when you run the quality control metrics, you may get vastly different absolute values from one scanner than another. And it's because of the, the proprietary underlying format that they've used to create that data set. And So it becomes difficult to compare across scanners, for example. The quality metrics are not not easily comparable.
Do we have any other questions for our speaker? If not, let's thank her again. And thank you, Diane. This was wonderful. Everyone, we will see you next month. Get those grad student abstracts in. I'm excited. And we will choose a time based on the 3 selected. So, okay.
Great.
Thank
Thank
you
you,
very much.
everyone.
It was fun.
Thank you.
Thank you, Diane.